Hi dear students, today we begin our lesson with respiration. Respiration is the oxidation of food to release energy. Respiration are of two types, aerobic and anaerobic respiration. Aerobic respiration occurs in the presence of oxygen and provide more energy. Anaerobic respiration takes place in the absence of oxygen and provide less amount of energy as compared to aerobic respiration. Respiration is a process in which organic molecules act as a fuel. Organic molecule may be carbohydrates, proteins, or fats. The organic molecules are broken down in a series of stages to release chemical potential energy, which is used to synthesize ADP. The main fuel for most cells is carbohydrates, usually glucose. Glucose breakdown is divided into 4 stages. Number 1, glycolysis. Number 2, link reaction. Number 3, Krebs cycle. And number 4, oxidative phosphorylation. We are going to explain each and every one of them in more detail in the coming lessons. Let's begin with the glycolysis. Now, dear students, look at here. Glycogen or starch. These are complex carbohydrates. Those are broken to release a simple sugar called glucose. Glucose is converted to pyruvate. Glucose is a 6-carbon molecule and pyruvate is a 3-carbon molecule. Now, 6-carbon molecule is divided into 2 molecules of 3-carbon. Pyruvate is a 3-carbon molecule, so 2 molecules of pyruvate are produced. This reaction is said to be glycolysis because glucose molecules split up to form 2 molecules of pyruvate. Okay, and they produce, now pyruvate is converted into acetyl coenzyme A. This reaction is said to be the link reaction. Okay, and acetyl coenzyme A enters into Krebs cycle. Okay, now glucose is not the only molecule they can enter into Krebs cycle. Protein molecule can also do so, and lipid molecule can also do so. Proteins are converted to amino acid into acetyl coenzyme A. Lipids are converted to fatty acid and fatty acid into acetyl coenzyme A. Acetyl coenzyme A enter into Krebs cycles. Okay, we are going to study all these steps in more detail in the coming lessons. These students, look at this structure. This is a typical cell. This part of the cell is said to be cytoplasm. Cytoplasm is actually the space between the cell membrane and the nucleus. Okay, this whole structure is said to be the cytoplasm. Cytoplasm is the site for most of the chemical reactions of the cell. Okay, glycolysis takes place in the cytoplasm of the cell. Glucose molecules are converted into 2 molecules of pyruvate in a series of steps, and this is said to be the glycolysis. For example, the first 2 steps of glycolysis requires energy. For example, glucose molecule is converted into fructose phosphate, which is a 6-carbon molecule, and an ATP molecule is used. Again, fructose phosphate, which is a 6-carbon molecule, is converted into fructose bisphosphate, and this also requires energy. So the first 2 reactions are endothermic. It means that energy is provided from the ATP. 2 molecules of fructose is converted into 2 molecules of triose phosphate, which eventually convert into 2 molecules of pyruvate. This is a complete reaction. Okay, and in this chemical reaction, glucose molecule is finally converted to 2 molecules of pyruvate, and this is said to be the glycolysis. Okay, now when pyruvate molecules are produced, what happens? You can see there is a structure called mitochondria. These structures are said to be mitochondria, and we studied mitochondria in detail at the beginning of these lessons. We studied that they are surrounded by 2 membranes, the outer membrane and the inner membrane. So pyruvate molecule enters into the pores or into the mitochondria through the tiny pores into the intermembrane space, and then the mitochondrial matrix. Now the reactions take place inside the mitochondrial matrix. Okay, now let's begin with a new lesson. Okay, glycolysis. Glycolysis is the splitting or lysis of glucose and takes place in the cytoplasm of the cell, as we discussed. It is a multi-step process in which a glucose molecule with 6 carbon atom is eventually split into 2 molecules of pyruvate, each with 3 carbon atoms. Energy from ATP is needed in the first steps, but energy is released in the later step when it can be used to make ADP. There is a net gain of 2 ATP molecules per molecule of glucose broken down. Okay, just because this is also this step of respiration does not require any oxygen, therefore they produce very smaller amount of energy. Okay, next, the link reaction. What is link reaction? Link reaction means pyruvate molecule are converted into acetyl coenzyme A. This chemical reaction is said to be the link reaction, but here I would like to explain 2 more terms: decarboxylation and dehydrogenation. Decarboxylation means a removal of carbon dioxide gas, and dehydrogenation means a removal of hydrogen gas. So from pyruvate molecule, carbon dioxide gas are removed and hydrogen are removed, and acetyl coenzyme A is produced. Okay, pyruvate passes by active transport from the cytoplasm through the outer and inner membrane of mitochondria and into mitochondrial matrix we studied in the diagram. Here it is decarboxylated, it is not carboxylated. Okay, it is decarboxylated, I forgot. Okay, you add decarboxylated and dehydrogenated and combined with coenzyme A. This is known as link reaction. Okay, pyruvate is decarboxylated, carbon dioxide gas is removed, dehydrogenated, hydrogen gas is removed and combined with coenzyme A. This is known as link reaction. Coenzyme A is a complex molecule composed of nucleoside adenine plus ribose with a vitamin pantothenic acid and act as a carrier of acetyl group to the Krebs cycles. Okay. Now look at here. You can see pyruvate molecule is converted into acetyl coenzyme A. This chemical reaction is said to be link reaction. Now what happens? Hydrogen gas are removed, so dehydrogenation. Hydrogen is removed, dehydrogenation, carbon dioxide gas removed, decarboxylation. So pyruvate is dehydrogenated and decarboxylated to produce acetyl coenzyme A, and this reaction is said to be link reaction. The Krebs cycle is a closed pathway of enzyme-controlled reaction. Acetyl coenzyme A combined with the 4-carbon compound oxaloacetate to form 6-carbon compound citrate. The citrate is decarboxylated and dehydrogenated in a series of steps to yield carbon dioxide gas, which is given off as waste gas, and hydrogens, which are accepted by carrier NAD and FAD. Oxaloacetate is regenerated to combine with another acetyl coenzyme A. Okay, dear students, Krebs cycle is not simple as I have explained them in one single slide. It requires several steps, almost 8 molecules. We are going to study them in the next lesson. For now, that's enough. Thank you.